Hey everybody, I'm Matthew Howell and I'm one of IMCS research scientists. I graduated from Wofford College in 2017 where I completed majors in biology, chemistry, and mathematics. I went on to Yale's Microbial Sciences Institute to spend 2 years with Sterula Hatsios looking at the post-translational processing of a Vibrio cholerae protease. I went on to do a PhD in chemistry at the University of Connecticut where I worked with Alfredo Angeles in looking at the activity of a family of tunicate antimicrobial peptides. I joined IMCS in 2024, the TIPS R&D team, where we work at developing chromatography products to be used on automated liquid handlers. Dynamic Devices is the only platform that will let us do up to 24 samples simultaneously. With 5 ml pipette tips, you can load an amount of resin that will let you handle 500 to 1,000 microliter sample volumes. With buffer exchange and desalting applications, we can load a very large sample volume relative to the amount of resin that we actually pack in a tip. So this kind of workflow is perfect for IMCS's tip-based separations. Let's view a demo of IMCS SizeX 1000 on the Dynamic Devices Lynx LM1200 using their 24 standard technology pipetting tool. In about 15 minutes, we will remove a small molecule contaminant, that is cobalt 2 chloride, from an aqueous solution of green fluorescent protein. 24 1 milliliter samples will be processed in parallel. For this demo, we use 3 boxes of 5 ml tips from Dynamic Devices, 1 box of SizeX IMCStips filled with inert buffer exchange resin, 2 24 well plates, and 2 troughs, 1 for PBS and 1 for waste disposal. We begin by dispensing air through the tips to remove the storage buffer that preserves the shelf life of our product. We then enter an equilibration phase where an amount of buffer determined to remove all trace storage buffer from the resin bed is added. More time is spent in this phase of the workflow for SizeX 1000 compared to SizeX 750 because there is less volume above the resin bed to add the required amount of buffer. Next comes the sample loading step, where reverse pipetting is here used to minimize sample loss during transfer. We next dispense air through the SizeX tips to drive the full volume of sample into the resin bed. A final volume of buffer, that is, chaser, is then added to the SizeX tips prior to dispensing air through the tips to complete sample elution. Whether you're working with smaller proteins such as GFP or larger molecules like polyclonal antibodies, SizeX tips reproducibly give efficient sample recovery. The same has been demonstrated for megadalton targets, such as plasmids and viral particles. IMCS provides generalized scripts that give excellent purification over this large size range, allowing the end user to immediately begin using our products. Tailored solutions for specific needs can be provided on request. Whether you're desalting following ion exchange or removing imidazole after nickel iMac, SizeX on Dynamic Devices has you covered for rapid, high-throughput sample purification. SizeX can be coupled to other IMCS tips applications, such as an affinity workflow, to transform manual separations into fully automated methods. Give the product a try and start saving time today. If you have any questions about our SizeX products for Dynamic Devices, or would like to request a free sample, please reach out to our technical support team. You can visit our website at www.imcstips.com, email us at inquiries@imcstips.com, or call us toll-free at 1-888-560-2073. Thank you.